My first question is, how long have you been a scientist? And when you started, what were the goals that you wanted to achieve through your research?
So I guess it depends on how you define scientist. I think I had an interest in being a scientist since I was in high school.
Hmm.
Um, so so basically, going back, when I went to college
Hmm.
as an undergraduate, I started to spend a lot of time working in a lab, and decided that that's really what I want to do. So we're talking going back, I guess, around till the late '70s.
Oh, wow. Cool. Do you have like a like a turning point in your career, like a defining moment in your career so far as a scientist?
Yeah, I don't know that I had a turning point. I mean, I think there are steps, there are areas that one is interested in studying, and questions that
Hmm.
one is interested in pursuing. And then there's a certain amount of uh, just happenstance that you end up
Hmm.
coming across an interesting problem, and you end up pursuing it, which may lead you astray from where you started. So
Hmm.
I guess one of the things that I've had an interest in, sort of going back, um, close to the beginning, was viruses, and basically how viruses replicate, and how they can um, basically how they can be used to manipulate cells.
Hmm. Cool.
And so I started off probably as somewhat more of a biochemist, and then
Yeah.
I moved towards neuroscience, and now I've moved towards oncology. Um, so I sort of pursued things that seemed interesting to me,
Hmm.
and that I thought I could make a contribution.
Nice. So, have you ever gotten like what were your most important results? If that's okay with asking, like it's along the same line, but I'm really interested in
Yeah, yeah. So
especially.
things, yes, there's lots of, I guess, they always say, oh, what was the aha moment, or whatever it is.
Mhm.
But there's, I think one of the most satisfying steps was basically developing a virus and then taking it into patients. So seeing
Oh,
the
yeah.
first patients treated with the virus was really
Oh,
kind of
wow.
an exciting and kind of a little bit scary
Oh, that's so cool.
highlight, I think. So that was really that was really great, and I think in a general sense, another highlight, although not directly relevant to my work, although we
Mhm.
contributed to its goal, was when an oncolytic herpes simplex virus was recently approved for clinical use, and so
Oh,
all of a
wow.
sudden, that was about a year and a half ago, and so all
Mhm.
of a sudden that opened up all sorts of new opportunities and
Wow.
interests from people in pharmaceutical industry and in
Yeah.
venture capital and things. So that
Mhm.
was another interesting step in the progression. But there's just every time an experiment works really nicely and you learn something unexpected, it's kind of that
Yeah.
that's real highlight. So um,
Mhm.
so some of the more recent things we've done, I think, are we have been really thoughtful, innovative, and interesting studies that have provided new insights. And so,
Wow.
like everything else, I guess the most recent things always probably tend to be the most exciting at the time.
Mhm. How do these like oncolytic viruses do they fare better than with like the standard practices for like treating cancer now? Or
Yeah,
like,
so they're. I mean, one of the interesting things about them is that they're really totally different.
uh-huh.
They really don't relate very well to any of the sort of therapeutic strategies. So, you know, so a lot of cancer therapy historically has been directed towards obviously killing the cells, but
Yeah.
through the use of chemotherapeutic agents, for example, so toxic
Yeah.
drugs, and then through the use of radiation, which is another
Mhm.
not a drug, but basically a toxic agent.
Mhm.
And then more recently, there's been this desire to kind of target things more selectively. And so there's so called molecularly targeted drugs or
Yep.
these small molecule inhibitors. Um, so the nice thing, and some ways even scary thing about the virus is um, is we're sort of just unleashing this biological agent in this tumor, and trying to get it to do things using its own basic attributes, biology, whatever you want to call it. And so I think there's many advantages to that, because we're kind of harnessing the biology of the virus, the ability of the virus to sense the physiology of cells, and just overall, it's really incredible potential to kill things that that it I don't want to say likes or dislikes, but basically that are permissive for it. And then I think that the side is that this somewhat scary side is that there's a lot to the biology that we don't fully understand. So we don't really have a specific target. Like, you can't say that, like, we know that radiation therapy basically induces breaks into DNA, and that's toxic to cells. It induces reactive oxygen species, which is toxic to proteins in cells. The virus does a lot of things, but it's hard to basically put or ascribe any one feature activity of the virus to the overall efficacy. And then the other thing that we would kind of interested us right from the beginning was this idea that in order to kill, the virus was actually replicating. So these are replication competent viruses.
Okay.
So it sort of means that if you put in a dose of so-called drug, the virus, that with time it should amplify itself within the tumor. So contrary to almost every other drug that basically decays or degrades with time in the patient or whatever, this had the potential to actually increase, expand, amplify in time. And so you had the ability of this sort of biological agent to kind of spread throughout the tumor, hopefully in a fashion that you didn't have to keep treating the patient constantly. So you're basically letting the biology of the virus do its work. And so that's the sort of optimistic view of it, or the hypothetical view of it, because obviously, as you're probably familiar, you're just looking at virus ecology, there's a constant battle between the host and the virus. And they evolve different ways to make themselves effective, and then the host obviously develops ways to block the virus. And in the same way, there's a sort of back and forth between the patient, the tumor, and then the virus, and the idea of controlling the virus, whereas the virus interests, or we try and alter the virus so its interest is to spread and kill the tumor cells. So there's a lot going on.
So then, if the virus never actually like dies and keeps replicating, would that mean like there would be no reoccurrence of the cancer technically? Because like the virus targeting those cancer cells would still be,
right. So it's a good question, and it's there's no good answer to it. One thing I can say is, obviously, for that to be effective or active, there has to be some reservoir for the virus to remain. And then it has to be able to in some way find those recurrent cells. But the idea is yes, you could have, for example, some sort of smoldering infection that as the tumor cells started to grow again, that those would then be targets for the virus. But it's probably not quite so simple. And we think that probably a really important component of this is, rather than having the virus spread and directly kill or infect these recurrent tumor cells, that the virus is actually inducing an immune response. And that immune response then is capable of clearing recurrences or cells that have potentially mutated or whatever it is. So the idea is that or at least the idea currently is that it's sort of a two-pronged approach, and that if the virus is working effectively and can generate this anti-tumor immune response, then that may be sufficient to then actually manage metastatic sites of tumor growth or recurrences of tumors. So that's kind of the hypothesis or the goal we're trying to achieve.
That's so cool. Well, where do you see this field moving in like 20 years? Do you see it as possibility that like these could become a staple for cancer treatment?
Yeah, so that yeah, I think so. I'm an optimistic person. I mean, yes, I think it will become a new sort of class of therapeutic agents, um, and um, and I think that it has a great deal of potential. Yeah, I see it growing in the next
Mhm.
couple of decades.
Nice. And do you have any advice for like me or someone else who wants to pursue something like this?
Um, well, I think the advice would be jump in. So obviously, there's different stages to everyone's career, but so one thing, obviously, is you would have to, it, I think first step is obviously learning about the field or what seem to be interesting questions to you, and then trying to find a laboratory where you could actually learn how to do some research and do some research because there is quite a gulf between reading about things in a textbook or even in the papers, and then actually sitting in a lab and doing the experiments.
Mhm.
And so I think you want to really one wants to move to a place where one can see if one really enjoys working in a lab doing research. Presumably, you're finding that out now.
Mhm.
Um, and then the question is how how you can what are the important questions to you and how to go about trying to address those. And so obviously there's a whole learning phase of this. There's a training phase of this. There's becoming an independent investigator phase where you have a lot more latitude in what you're selecting or what you're working on. But um, I think that there are multiple ways to get involved. But
Mhm.
I think you know for me the underlying excitement has been doing the research, so to speak.
Nice.
And so I think that um, there's a lot of interesting things that can be done. And the other side now even is people can there's a lot of this is moving into the biotech industry and the pharmaceutical industry. And so one doesn't have to necessarily to pursue this stay in academics. There's, I think, many more opportunities now for people to be involved either from the business standpoint or even the research standpoint, in companies that are developing these therapies and testing the therapies.
Mhm. I guess my final question is then, well, how come you chose academics? Maybe then, like all the other routes you could have taken.
I probably wasn't qualified for anything else.
No, yeah, I mean, yeah.
I mean, so there's, it's just, it's something that's interested in me, as I said, for a long time, from basically from high school.
Mhm.
Um, and so, I mean, there's a lot of things I like um, I like in some ways the independence or the ability to set my own course.
Mhm.
I like the ability to have my own flexibility in what I do and when I do it. Um, I like the intellectual aspect of it. In other words, there's always something challenging and thoughtful and new. One is always learning new things.
Mhm.
Um, I like the interaction with other scientists. I think the whole give and take and the ability to be critical without being personal, to really try and delve deeply into things and to question things. I guess I like to question things going back a long way, and so this is an avenue where sort of um, something that's expected is to ask critical questions and without being too concerned about making it or viewing it as something that's antagonistic.
Mhm.
Um, and and I think it's also really great seeing so-called students, younger people, trainees, and just their excitement, their enthusiasm, and trying to convey and teach some of the things that I've learned over the years.
Mhm.
So that's a hodgepodge.
No, thank you. Yeah, those were all my questions. Thank you so
Okay.
much for your time. Yeah, I really enjoyed it.